So we're really excited. Today, our speaker is Amanda. Amanda has a master's degree in agricultural life sciences with a concentration in biochemistry and a bachelor's in biochemistry and molecular biology from Mississippi State University. Their research experience is largely in using molecular biology for understanding of host parasite systems in amblyomma ticks. Then their masters explored microRNA expression of maize and aspergillus using small RNA-seq. And since joining Novogene in October 2019, Amanda has been excited to be a part of our team improving the customer experience with Novogene. So we're excited to have Amanda present this webinar for all of us today. So with that, Amanda, I'd like to hand it off to you, and yeah, go ahead and take it from here.
Okay, thank you, Sippy, for the introduction. Again, I'd like to say on behalf of everybody, all the moderators, thank you all for coming today and coming to hear about our best practices for preparing your samples to come to Novogene. So briefly, just a little overview of what we're going to be talking about today. I'd like to start with just a brief introduction of Novogene, the company, then some of the steps that we need before you can start to submit samples, followed by a couple of details about sample preparation that we'd like to share with you guys, followed by preparing your shipment, finally. The most important part, just a little overview of our sample check-in, and then we'll do a brief review, and then we'll go to the Q&A. On behalf of myself and Dr. Novogene here, and the technical support team, I'd like to say thank you again for coming out today. So again, briefly, our founder-in-chief, Dr. Lee, is a very accomplished researcher, developed a software, best known for developing the software SOAP, which is used for bioinformatics. He was previously the vice president of BGI and then ended up moving forward to found his own sequencing company in Novogene. That founding occurred in 2011 in Beijing. And in the almost 10 years now, since then, we're now a global sequencing and bioinformatics servicer for both research and clinical types of samples. We do have one of the world's largest sequencing capacities, and we are working with the latest Illumina, PacBio, nanopore sequencers at our facilities worldwide. And one more time, just to stress, we are a global NGS provider. On our website, which I've got a link here for you guys if you want to look at it later, you can find all sorts of information about all of our different labs globally, as well as all of the different services that we offer. We've been updating the website quite a lot, so it's looking very good right now. And over here, we just have some photos of the headquarters in Beijing. I haven't had the pleasure of going yet, but one of our moderators, Mary, has. She was there last winter, and she said it's very nice. So now, step one in getting your samples to Novogene is to request a quote. If you have contact information for your regional sales representative already, or a technical support, or basically any Novogene employee, you can shoot anybody an email or a phone call, and we can get you in touch with your regional sales representative. Or if you've never worked with us before, you're absolutely welcome to visit our website. We have a module where you can contact us, give us a bunch of information about your project, and then request a quote there. Those requests go automatically to the appropriate salesperson, and they can get in touch with you really fast. At this point, we want to hear everything about your project. We want to hear all the way down to where the research question comes from, any background data you have. We're here for it. We want to hear everything about the great research that all of our clients are doing out there. I know, so my territory is Boston. I do technical support for that area, and there's so much great cancer research going on. I love to hear all of the interesting work and groundbreaking work that everyone is doing. Um, Obviously, besides that, we do need some more tactile information, right? Sample number and where the samples are coming from, what types of samples you're looking to send to us, either nucleic acids, libraries that you've already prepared, or tissue that you need help with extraction. And finally, we definitely need some biosafety information, okay? At this stage, we want to ensure that we can do the work that you're looking for. And obviously, I've bolded it here because this is something that we would like to stress to all of our clients who are new and returning. So I'm actually going to read this one. So our global labs operate under global, national, and state level standards, including laws about what can be transported across state or national lines. So we are sort of juggling a lot of different rules as far as safety goes, as far as, even if it's like endangered species or things like that. And because of this, each individual project that you work with us on is going to be evaluated to ensure that we can legally and also safely handle your samples. So again, just to stress, even for extracted nucleic acids or something that you might not consider to be a biosafety issue within your lab, we're going to definitely need as much information as possible. Again, even for things that you might not consider, we're going to need as much information as possible to make sure that we keep anybody who might handle your samples safe. And again, let me just stress this. This isn't about, turning any projects away. It really is, we want to know everything that you're doing, but we also definitely need to know that we're going to be able to legally and safely work with it. Once you have your quote, you can see down in the bottom right corner, this sort of letterhead on the top of your official quote. I've put a red circle around your quote number here. This is just an example, of course. That number starts with NVUS, so Novogene US, in case you're interested for the origins of that. That number is your best way to communicate about a specific project. So once you have a quote number, any communication about your project is going to probably include that. That's our easiest way to search your project in our databases and our systems and make sure that we all know what we're talking about. Once you have your quote, the next stage is going to be confirmation of payment. Generally, requesting a purchase order from your institution is sort of the go-to for most of our clients. However, for a lot of international clients, especially, prepayment via credit card or wire transfer is also an option. This is the step that we would want to take for initiating your project. This is how we know that you're serious, and how you know that we're serious. We enter into this contractual agreement with you. Following the project initiation, I would say, if you haven't already, prepare your samples. As you work to prepare your samples, we do have a download for under our resources tab. We have several downloads, but we have a download for all of our sample requirements. It's quite a large PDF document, so I've just pulled one here just as an example for today, for eukaryotic RNA sequencing. As you can see, looking at the first row, we are looking for total RNA from, if you're working with animals, for example, and we're looking for at least 0.4 micrograms and a volume of at least 20 microliters and a concentration of 20 nanograms per microliter. We're looking for decent RNA integrity. Or if you don't have the access to an adjuvant, obviously using a nanodrop can also help you get an idea of your RNA purity and things like that. This is just an example of one. Obviously, for every single product we offer in that document, we have a list of the different sample types that you could be submitting and all of the specific requirements. This document has been worked on by our product specialist team. And it's based in statistical analysis on what we know that we can guarantee for you. And this is the good news about working with Novogene, is that we do, if you're meeting our requirements, we do guarantee your sequencing results and output. Outside of this, I would like to mention here that we do offer some lower input options. Say for RNA sequencing, for example, we can sequence samples that you would submit below 0.4 micrograms total. Some of those are doing work with other labs, and some of them do have different pricing points. So in order to meet your different projects' needs, I'd recommend talking to your technical support. If you are preparing your samples and you're finding that you're not getting the appropriate amount to meet these requirements. We definitely do want to help. We want to work with all sorts of different clients and all sorts of different samples. So we do have other options outside of this very general set of requirements. Let's see. So you've prepared your samples. Everything is ready. Hopefully you shared your sample information with the technical support team. I'd love to push for you to do it through our CSS customer service system. If you haven't been invited, I'd definitely, ping your technical support and ask them to invite you right away, because it's going great right now and working finally. We're so happy about it. So preparing your shipment. Over here on the right side, I have one of our many pre-shipping checklist type documents. It has a handful of just bullet point sort of steps to complete before you're ready to send your samples to us. To summarize this, I'd say there are sort of 4 important elements to preparing your package. The first element is your tubes. This is the cradle where your samples will be contained while they're in transit. Very important that they stay inside of them. We definitely want to have either a 1.5 or 2 milliliter flip cap style tube. So I've enlarged the picture from the document right here in the center, so you can see exactly what I'm talking about. Obviously, incorrect tubes mean that they don't fit into our automated pipelines, and so we're going to have to transfer them to new tubes, and that will usually mean that you are going to be charged $5 per tube. So I would definitely recommend a 1.5 ml flip cap tube. On this tube, I need you to label it with a black permanent marker on the top and on the side. This makes sure that if the top label gets erased, we still have the side label, or vice versa. And also Novogene is going to add our own label on top of it. So having both of the labels, everybody's labels everywhere, we all know what we're looking at here. And nobody's samples get mixed up. As far as that labeling, it needs to be no more than 8 characters, which I know I've worked on projects that have had so many different elements that we've had so many different aspects to the name that they've gotten to be upwards of 20 characters before. However, can you really very clearly write more than 8 characters onto the top and the side of the tube? It's very difficult without having a printed out label. And we don't want the printed out labels or any sort of stickers or tape. This will sort of prevent the tubes from fitting in the centrifuges. Again, a lot of our pipelines are automated. This is what helps us get our turnaround time within what we have we've quoted you. So having those sorts of barriers is definitely going to prevent that. So this is our best way to ensure that we're reaching your turnaround times and making sure that nobody's samples get lost. Everyone's samples are exactly what they say they are. So the tube. Next, the packaging. And obviously we would want you to put your samples inside of the tube. I feel like that's probably self-explanatory though. Onward to the packaging. So now you have your tube or tubes. If you have greater than 24 samples, we are definitely going to ask that you put them into a freezer box or something that sort of has the dividers in it so that you can put the tubes in order. We can very easily, our sample receiving team, I should say, can very easily look over, make sure all of the names match the names that you've shared with us, and move them on to being checked in very quickly. If you have fewer than maybe 10 samples and you think they would fit into like a 50 ml conical tube, that would work. We'd recommend putting something inside of the conical tube, like Kim wipes or something like that, to sort of protect them as they're being jostled during shipping. Let's see, I have a few notes. We recommend putting both the boxes and the conical tubes into Ziploc bags, just in case something were to happen, the box were to rip, the conical tube were to break. We want your tubes to be inside of something safe rather than floating around inside of the box in with all of the dry ice. And again, this is something that seems very self-explanatory, but maybe it's a good idea to also put a rubber band around the box to make sure that it stays closed so that your tubes stay in order. All of these sorts of secondary precautions are the best way that you can keep your samples safe. And look, I went and forgot something. As far as the tubes, let's go back to the tubes briefly. That third point you can see written in red, parafilm prevents leaks during transit. We also recommend wrapping some parafilm around the lid of the tube, around the outside, of course. And as you saw on the previous slide, our sample requirements, the volume requirements, 20 microliters is not a lot of volume. If it gets a little warm, maybe that liquid would evaporate, catch on the lid of the tube. If the tube comes open or something happens, even slightly open, you could lose some of your sample. And so parafilm is going to be the best way to prevent that from happening. Again, back to packaging, I think that I've hit the high points. Put your tubes into a secondary container, inside of a Ziploc bag, inside of your dry ice. And this is something else that our sample receiving team has actually asked me to stress to you guys. We don't want dry ice in with the tubes, right? So inside of the box, we want to keep your samples separate from the dry ice. I know that, it makes sense to maybe want the dry ice to be closer. Maybe it keeps everything cooler, but your best bet is going to be keeping the samples in 2 or 3 layers of packaging inside of the dry ice and keeping sort of the exterior environment cool instead. Speaking of dry ice is the third very important element. So we recommend 5 pounds of dry ice per day. This is just based on our general understanding of how quickly it sublimates, and 10 pounds for an overnight shipment. So generally, you're going to want to put 10 pounds. However, more dry ice can never hurt. Another safety precaution that we would add is shipping your samples earlier in the week is your best way to prevent them getting stuck at a FedEx facility over the weekend or stuck at a UPS facility over the weekend. Boxes that don't get delivered do sit in a warehouse outside, and if you're coming through Sacramento gets quite warm, especially during the summer. Even in the winter, it's not really that cold. It's not dry ice cold at the very least. So dry ice, very important, a great way to prevent your samples from degrading, especially RNA. RNA has to have the dry ice. And regarding dry ice, one of the best ways to make sure that FedEx or UPS or whoever your courier is knows that your samples do need to be cold is to make sure that there's a dry ice label on the box somewhere. That's something that we actually see less and less of nowadays. And the SRT sample receiving team has asked me again to stress that. Having that, it's the black label in the diamond shape, that diamond label for dry ice. It will inform the courier, whoever is moving the package, that whatever's inside needs to be kept cold, and it's important that they deliver it in a timely manner. And finally, the fourth and final element to ensuring that your package is prepared for shipment is to include your packing slip. The technical support team is generally the one who provides you with a packing slip. Also, if you are on CSS, CSS does allow you to enter your sample information and then print out a packing slip right then and there. Again, I can't stress this enough. I highly recommend CSS right now. Please make sure that you put it in the box. The CSS packing slip have a number. So as of this date and time, they don't have a barcode, but they do have a number that starts with SUS. That is a unique identifier. And within our system, we can access your sample information immediately as soon as the samples arrive. We have other barcodes that are other packing slips that have barcodes on them. However, there are occasions where your technical support would ask you to fill out a sample information form and just include that. Please include that. If that's what they've asked for, include whatever it is that they've asked for. If by some very strange circumstance you can't include any of those things, please do write the NVUS number somewhere that we can find it. And again, like I said before, that NVUS number is your project. As far as our system goes, it will be the easiest way for us to figure out who the samples belong to, which technical support needs to help, etc. So you've prepared your shipment. Anyway, I want to go over a few more do's and don'ts. Again, this is what you guys are here for today. Just a reminder, so at our Davis lab, if your samples are going to stay there or going to our Tianjin facility, they're going to go through Davis. So the sample receiving team there receives hundreds of tubes every single day. And in order to make their job a lot easier, we're asking you to follow all of the rules in the checklist. This is, the checklist is for them. It's for all of us, but it's for them primarily. And this is our best way to sort of make their lives a lot easier. And honestly, they are, so maybe you guys don't know, they do have two different individuals checking over to make sure all of your sample information matches. So they are doing their very best to make sure that everything is correct before we even start doing quality control checks or anything. So again, I believe I mentioned the first don't here at the bottom left. Please don't put dry ice into the container with your tubes. You can see an image here where the sort of dry ice pellets have been put into the cardboard dividers. And so the empty space would have been where the client's tubes were. Again, this is sort of a mess to deal with. Dry ice isn't really something you are safe to touch anyway, so we don't really want to be touching all over it. The next one is an image that one of my clients did recently, putting parafilm inside the lid of the tube. The point of the parafilm, in theory, is to be wrapped around the outside to ensure that the lid stays as closed as possible. And having it inside of the tube, I would be concerned that some of your sample would end up on the parafilm, and therefore it might get thrown out. And finally, another image with packing peanuts. Honestly, packing peanuts are quite a mess, and maybe pulling from some stereotypes, but California is generally very green. We do like to recycle as far as, I'm in North Carolina, but I think that they prefer to be able to recycle things. Packing peanuts can be quite a mess, and they're sort of hard to clean up and deal with and dispose of in a green fashion. And also, they've requested that we don't include real ice, so ice made of frozen water. The major reason here is that real ice turns into water, and water can leak out of the foam container that your samples are in. And if it leaks out, it will damage the cardboard of the box. If the cardboard of the box is damaged, then it adds another step for our sample receiving team, just making sure that they're not being put at risk, that none of your samples have gotten out of their containers and things like that. It's all about safety here with that. And honestly, that cardboard box is that final layer of protection for your samples from the outside world, so we don't want it to be destroyed. So you've shipped your samples. If everything has gone absolutely perfectly and you've done your best to prepare your shipment correctly, your samples get checked in automatically. We will use that barcode and get your sample information almost immediately. And you can receive confirmation the second that your samples are checked in, if you have your CSS account set up. So here, Dr. Novogene is very happily saying, good job. Here below that bullet point, you can see an email example. So dear customer, we've received your samples. That SUS number, again, is that batch ID that we're using to identify them. And then you can proceed with quality control, or we'll proceed with your quality control testing, and we will, again, notify you once that's been complete. You get that email literally automatically. I know because I'm copied on all of them. However, if your samples do have any issues, maybe there is an issue that the names don't match those names that you shared with us, or there's an issue with the packaging. Say your real ice melted a little bit and now the cardboard is sort of not integrity anymore, or something to that nature. Your technical support will get in touch as soon as we've figured out what's going on and who the samples belong to, of course. When they get in touch, they'll remind you of the correct packaging protocol if that's applicable for you. And your samples will be checked in as soon as we have the information that we need, maybe the new namings or something of that nature. This does mean that your check-in will be delayed. So I've made that note bolded again too here. A delay is not necessarily bad. It just means that this is additional time added to your turnaround before your turnaround time actually starts. So this is especially for those of us who have who are looking for very quick data return. This might make or break how quickly you get your data back. So that's it, you guys. This is it. You're done, right? So again, Dr. Novogene and I are here to think about, so how exactly do I get my samples to Novogene for sequencing? Step 1 is to request a quote. We'll confirm payment and initiate your project. If you haven't already, start to prepare your samples. And then once your samples are ready, all your sample information is to us. You start to prepare the shipment. Once it arrives in Sacramento, we will do your sample check-in, and then your samples move forward to quality control or extraction. At that point, you can always, we're gonna stay in touch. Your technical support and your sales team will stay in touch. We usually pass your project along to your project coordinator at that point, and that's when library prep, sequencing, data analysis starts. This is the good part. So I'd like to say thank you all again. I'd like to say a personal shout out to my supervisor who recommended me for this presentation. I'm very thankful to be here. And again, thank you all for your time today. And so I think we're ready to move to the Q&A portion. Sibi, did you want to introduce the next couple of webinars before I start? Yeah,
absolutely. So thank you, Amanda, for helping us walk through this webinar. Thank you for the valuable information. As Amanda mentioned, we are now in our live Q&A portion of this webinar. If you do have any questions, please do use the Q&A module to post it there. We're going to give it a few more minutes to let some questions populate in there so that we can start answering that. We have a few minutes for that. But while that's going on, please do follow us on social media. You can find us on LinkedIn, Twitter, Instagram, Facebook, and you can use Novogene America to find us. To stay up to date on our latest news, promotions, any sort of contests where you can win sequencing vouchers and future webinars like the two highlighted here. The next one that's coming up in October and in November are NGS guides to human whole genome sequencing and epigenetic study of gene expression regulation as well. And so with that, let's jump into some questions here. Let's see. Amanda, we had a question from Heather. Did you mention where they could get those dry eye stickers, or is that something that they should reach out to their individual TS for once they're ready to ship and they can help coordinate with their shipping courier?
So as far as I know, I've never personally provided a dry ice sticker to someone. I just looked it up though. And so FedEx has one that you can just sort of print off. Again, it's a black and white diamond and it has some black stripes on it. And I think they just need to know how much dry ice is in there. And you can fill it out and print it out and sort of tape it onto the box from FedEx, or I think you can also purchase stickers online, but I'm not sure that we are able to provide a sticker directly to you.
Yeah, and I think that if anyone needs help with that when you're ready to ship, you can always reach out to your technical support specialist and they will be able to help you coordinate and find that to be able to help get your samples into us efficiently, correctly, again, as Amanda mentioned. Again, we're taking a few more questions. If you have any, please do post them in the Q&A module. Please also note that we will be sending out the recording for this webinar and the slides in a follow-up email after the webinar. So whatever email that you use to register or sign up for this webinar will be where we send those to, so kind of stay tuned. You should expect that after this webinar within a day or two. So thank you again for joining us. We do have another question, Amanda, from Suli. She just wanted to make sure we use the 1.5 mil cap tube for each sample, which in their case is sort of total RNA. That can be 10 microliters or so. That is correct, right?
Yes, so we're looking for like a flip cap, not a twist on cap, but otherwise that sounds like the correct tube to me without seeing it. And again, your TS team can always, if you want to send a link to the type of tubes you're purchasing or something like that, that your designated TS can, of course, help to answer that too.
Awesome. Again, I think the main message is we're here to help. I know that our clients love our technical support and the efficiency that they work with. So if you do have any questions or concerns that we're not able to answer or that pops up as you're prepping your samples, our technical support specialists will be the best place to start. And they will answer your questions promptly and be able to help you make sure your project is a success. We have another question, Amanda, asking whether or not dry ice should be under Ziploc or if it should be just covering the bag. How would you go about recommending a solution for that?
I mean, I think that it should be fine either way. In my experience shipping things, especially because it's more than likely going to end up covering the Ziploc bag at some point during transit. So I think if you go ahead and just place a Ziploc bag on top, that should keep the environment inside of the sort of foam cooler cool either way. Yeah.
Awesome. Cool. Well, that's pretty much all the time that we have today. If there are questions that we have not answered during this webinar that you might have put in the Q&A module, we will be answering that in our post-webinar email. Also, if you do have any questions after this webinar or once you get the recording, we would love to answer that. You can always send that to us via our social media platforms or using the inquiry_US at novogene.com email. Again, that's inquiry_US at novogene.com. But if you have the email for your dedicated technical support, you can email them directly as well. So with that, we want to thank everyone again for joining us. We appreciate your time. We hope this was helpful. Please do reach out to us for any questions you may have regarding sample prep, NGS in general, or any bioinformatic analysis questions. We would love to help provide some options and give you some information so you can make the best decision possible for your projects. Again, we do have 2 webinars coming up, one related to human whole genome sequencing and another looking into the epigenetic study of gene expression and regulation there. So we would love to have you guys join there. You can find information on that on our social media pages. So thank you again. Have a great day. We will greatly appreciate your participation this morning. Thank you. Bye-bye.